Okay, let's look at biotechnology, a quick overview of biotechnology. We could spend a whole lot of time on biotechnology, but we're going to do a quick overview and touch on some key points that you need to know. So, when we look at biotechnology, again, there's a whole lot of topics, but we're going to just touch on a few of them. We're going to talk about restriction enzymes, gene cloning, and recombinant DNA technology. We'll talk about gel electrophoresis and DNA fingerprints, polymerase chain reaction, DNA sequencing, and CRISPR Cas9. Those are the top topics in biotechnology. So we're going to take a quick look at some of those. So biotechnology is using living things for a particular purpose. So we are looking at how we can use DNA and living organisms to do things for a particular purpose. So DNA technology has kind of revolutionized biotechnology. Biotechnology has been around for centuries, but with the DNA and understanding DNA and DNA technology, we've come a long way since the discovery of the structure of the DNA molecule. So biotechnology has changed significantly in the past 50 years. When we look at recombinant DNA technology, what we're looking at is we are looking at taking DNA from two different organisms and putting them together into one organism. So taking genes from one organism and putting them into another organism. Typically, this is done in vitro, so in a laboratory. So in vitro means in a laboratory setting, a test tube. Anytime you do something in the lab to manipulate stuff, living things, that's in vitro, as opposed to in vivo, which would mean inside the living body. So some experiments are done in vivo, but biotechnology is typically done in vitro. So starting with restriction enzymes are enzymes that cut the DNA. So think of them like DNA scissors. Restriction enzymes work by finding a restriction site, and once they find that unique restriction site, they then cut the DNA at that particular site. The restriction sites are typically palindromes, so they're going to be DNA sequences that are unique to that particular restriction enzyme. So the restriction enzyme is going to search the DNA, and anywhere it finds that unique sequence, it cuts it. And it's always going to cut it in exactly the same way. And the pieces that it cuts it into are called restriction fragments. When they cut, some restriction enzymes are going to cut it straight and down across the DNA, and those will create what's called blunt ends. But the ones that are most useful are the ones that actually create sticky ends, and they cut it with a zigzag cut so that there's bases hanging over the edge that could then stick back together with other bases as long as the base pair rule fits. So what scientists will do is they will cut the DNA with those sticky ends and then stick it together with another piece of DNA that's been cut with the same restriction enzyme. And then we can use DNA ligase to bind those pieces together. And so in this diagram here, you're seeing a restriction enzyme that has a unique sequence. So here's our restriction site, and we've got GAA-TTC is the sequence. If you notice on the other side, we also have GAA-TTC in the opposite direction. So the sequence is 5' to 3' GAA-TTC. And then what will happen is the restriction enzyme will cut the DNA between the G and the A. So it's going to cut it here, and it's going to cut it here. Then these hydrogen bonds will break because they're not very strong. And so once this covalent bond has been broken in the backbone, and this covalent bond has been broken in the backbone, the hydrogen bonds break, and the two pieces of DNA separate. The bases that are hanging over the edge here those could stick back together with the complementary bases that are hanging over here. So we could actually put these two pieces back because the base pairs will match. And so we can do the same thing with another piece of DNA. So a gene from another organism, if we cut it with the same restriction enzyme, it's going to have these same sticky ends. Those sticky ends can match these sticky ends, so they stick together. And then we're going to use an enzyme called DNA ligase to seal those pieces together. And if you remember back when we talked about DNA replication, DNA ligase connected all of the Okazaki fragments together. We're taking that same enzyme from that natural process and we're using it in a laboratory to connect those two pieces of DNA together. And so DNA ligase is going to seal those pieces. And now we've got a gene from another organism, this gray area, inserted into the DNA from the first organism, the green area. So if we go back real quick, we started out with a green piece of DNA. We cut it with a restriction enzyme. We inserted a DNA sequence that had been cut with the same restriction enzymes, the same sticky ends on both ends. And then we're sticking those in between, the gray one in between the two green ones. And DNA ligase covalently bonds those pieces together and permanently adds that gene into the other piece of DNA. So DNA, because again, it's a universal molecule, we can cut it with the same restriction enzymes and the sticky ends will stick together, and DNA ligase enzymes will seal it and bind it together. And that is recombinant DNA technology, taking that DNA and sticking it into another organism. So we're using restriction enzymes to cut it, and then we're using those sticky ends to bind it together. What we do in recombinant DNA technology is we use those same restriction enzymes and we take a gene from one organism and we can put it into another organism. Bacteria are great organisms for doing this because they reproduce really, really fast. And so we can get lots and lots of copies of those genes very quickly. So bacterial plasmids are going to be used as cloning vectors. And a vector is really just something that can transfer something from one place to another. And so it's going to be a transfer vesicle to transfer a gene from one place to another. In bacteria, they have, as we know, one circular piece of DNA as their normal DNA. But some bacteria will also have extra pieces of DNA, small circular pieces of DNA, and those are called plasmids. And so those small plasmids are what we use as those transfer vesicles. They're great for this because they're small and they're easy to add genes to it, and we can sequence them really quickly. We can figure out what restriction enzymes will work on those plasmids, and we can put the genes that we're interested in into those plasmids and put them into the bacteria. The bacteria also reproduce really quickly, so we can get lots and lots of copies of the gene that we're interested in very fast. So here we can see a picture of a bacteria, and that bacteria is going to have the bacterial chromosome over here, and it also has a plasmid. If we take those plasmids out of the bacteria and we cut them with restriction enzymes, and we cut a gene of interest, so let's say it's a human gene that we want to make a copy of, we want either proteins from it or we just want copies of the DNA, we can cut that gene out with those restriction enzymes. As long as we're using the same restriction enzymes, the sticky ends will stick together, the DNA ligase will bind them together. We can then put that plasmid back into that bacteria. That bacteria is then going to reproduce, and it's going to reproduce with binary fission. So it's making exact copies of that DNA in that cell, and it's going to make clones of the genes. And so once you've got lots and lots of copies of the genes, you can do all kinds of things with it. You can take the genes out and you can study the genes, you can research it, you can do all kinds of things, analysis with the genes, but we can also get the bacteria to produce the proteins from that particular gene, and then we can extract and purify those proteins, and we can do all kinds of things with those proteins. So that's gene cloning, and it's the bacteria making copies of the gene. That is the process of cloning. So gene cloning is using the bacteria as a host to make copies of the gene that we're interested in. I'm not going to show this video. I will post a link to it in Schoology so you can go back and watch it on your own. So that's recombinant DNA technology. And let's go back real quick to that. Insulin is an example of recombinant DNA technology. If we take the gene, the insulin gene, the human insulin gene out of a human, put it into a plasmid, put it into a bacteria or a yeast, we can get those organisms to reproduce and make copies of that particular gene for insulin, and then we can get them to produce the insulin that we need. So insulin is an example of a product on the market. It was actually the very first FDA approved medicine using recombinant DNA technology to produce insulin for human consumption for people who are diabetic. So it's a way of producing medicine and other proteins and gene copies that we can do all kinds of things with. So recombinant DNA technology uses gene cloning to make things like insulin. Gel electrophoresis is another process where we can look at the DNA, we can study and analyze the DNA, and it is a machine, and we're going to do this later in the year. It's a machine that will cut up, no, this doesn't cut up the DNA. The restriction enzymes cut up the DNA, but it allows us to then analyze that DNA, creating things like DNA fingerprints. And so it allows us to study the sequence of the DNA and gene expression and the functionality of the gene. And it is a machine that basically separates the pieces of the DNA by size. So gene expression, or gel electrophoresis, lets us analyze and study the DNA. So looking at that gel electrophoresis process, it's a machine. It uses the properties of DNA to separate out the DNA molecules by size. And so what you're doing is you take the restriction enzymes, you take the DNA and you chop it up, add the restriction enzymes to the DNA in a tiny little vial, and then you can put that chopped up piece of DNA into the machine. And typically what you do is you add it to these little wells here. And so they're holes in a gel. And then because the DNA molecule is negatively charged, you're putting it into this machine that has an electrical current running through it. And if the DNA is negatively charged and it starts at the well over here, it's going to be attracted to the positive end of the machine because you've plugged it into the machine, into the wall, and the electrical current is going through it. There's a positive end and a negative end. So if DNA is negatively charged, it's going to be attracted to the positive end of the machine. And so it's going to move across that machine till it gets to the end. But it's stuck in this jello-like substance, and so the DNA has to move through the gel to get to the positive end. But the bigger pieces are going to move much slower than the smaller pieces. So the smaller pieces will move faster. So it's kind of like a race. And because it has to work its way through the gel, the little tiny pieces can get through all those little grooves much faster than the big pieces can. And so the gel is going to separate out all of those fragments of DNA by size, the smaller ones moving farther than the bigger ones. And this is called restriction fragment analysis because you're looking at all of the fragments and you're analyzing them. And so we can use this to compare different DNA molecules. And so when we're doing that, we're comparing those restriction fragments. And so we're looking at variations between the markings or the DNA fingerprint band patterns. And so we're doing restriction fragment length polymorphisms. That's a way of comparing the different size pieces. So one example of a use for this is if we're looking at the gene for sickle cell anemia. The normal hemoglobin gene, when you use a particular restriction enzyme, and this right here is the name of the restriction enzyme, it's DDI1. And if we use DDI1 to cut up that gene, it's going to cut it into a certain number of fragments. If you are looking at the sickle cell mutation, what happens is the mutation is in one of those restriction sites, which means it's not going to cut it in that particular location. So you're going to get less pieces of DNA, and it's going to give you a different band pattern. And so here in this first column or first lane, we can see the fragments that are being produced from a normal allele. And so we get these 3 band patterns from a normal person or normal allele. If somebody has sickle cell anemia, their gene is going to have 2 fragments instead of 3 fragments. And so we can tell if somebody has the normal allele for that particular protein or sickle cell. So we can do some gene analysis to determine if somebody has a sickle cell gene or not. That's one example of a use for gel electrophoresis, is determining particular genes for particular disorders. But there's lots and lots of uses of DNA fingerprinting, and the pattern that's created from gel electrophoresis is called a DNA fingerprint. And we can use it in forensics to solve crimes. So in this diagram over here to the right, this is a picture of a gel, and we're looking at a lane. The first lane here has the victim's DNA, and then we've got a specimen that was left at a crime scene, and we have DNA from 3 suspects. If the person was at the crime scene and they left that specimen, then their DNA fingerprint is going to match the specimens. And so if you look at this very first suspect, all of these band patterns match. So it looks exactly the same. So that person's DNA is the specimen found at the crime scene. So we can solve crimes using DNA fingerprinting. You can use it in medical diagnoses, like with sickle cell anemia, and all kinds of other scenarios, paternity cases, things like that. So lots of uses of gel electrophoresis. But the general gist of gel electrophoresis is you are separating out DNA by size because the DNA molecule is negatively charged and it's moving through the gel. Okay. Another biotechnology technique is polymerase chain reaction, also known as PCR. And PCR is when we are making copies of the DNA. So basically what we are doing is we are doing DNA replication in a test tube or in a machine. So polymerase chain reaction is producing lots of copies of a specific targeted section of a DNA. So you're not copying the entire genome, you're just copying a particular DNA. But there's a few things that you need in order to do that. But basically, you're taking a little test tube, you're putting the gene that you're interested in it into that test tube, you're putting all of the nucleotides in there, you're putting all of the enzymes in there, and then you're doing a hot-cold cycle that will then make copies of the DNA. The key to polymerase chain reaction is an enzyme called Taq polymerase. And so, as you know, you need DNA polymerase in order to do DNA replication. But because the machine goes through these hot-cold cycles, we need an enzyme that's able to withstand hot temperatures. And so scientists discovered hot springs have bacteria that can survive in high temperatures. So they have extracted this enzyme, this Taq polymerase enzyme, from those hot springs bacteria, and they use it in the machine to do DNA replication as the DNA polymerase enzyme. And so what you're doing is you're starting out with a sequence of DNA that you're interested in making copies of, and the hot-cold cycle in the machine, the heat will denature the DNA molecule, which means it's going to break those hydrogen bonds between those two strands. Once you've got the DNA separated, then you can start building a new DNA molecule using the base pair rule. You have to, in the machine, add primers because, as you know, DNA polymerase needs something to start with. So you would add a primer, which will bind on to that beginning section of the DNA. And then the Taq polymerase will build the new DNA molecule. So as it starts to cool, it will build the new DNA molecule, and it grows until it gets to the end of that DNA molecule. And so after one cycle, you're going to have 2 pieces of DNA. Then it goes through another hot-cold cycle, and it goes through about 90-something hot-cold cycles. And after that, you've gotten thousands of copies of DNA of that very first sequence. So in your test tube, you're putting all of the nucleotides that you need, all the A's, T's, C's, and G's. You're putting the primer in there, you're putting the Taq polymerase enzyme in there, and it will build new DNA molecules using that original strand as a template. So again, you're starting with a piece of DNA with a target sequence. You're doing the hot-cold cycle to make a copy of the DNA, and then it's going to do another copy and another copy, and so it's going to keep doubling and doubling each time it goes through that sequence. In the pandemic, you probably heard of PCR a lot because there's COVID-19 PCR testing, and what they're doing in this process is they're making copies of the viral RNA to test to see if you've been exposed to COVID-19. So the process is a little bit different because the virus, COVID-19, has RNA as its genome instead of DNA. So it's actually PCR testing for COVID-19 is actually reverse PCR testing. So what it will do is it will first make DNA from the RNA, and then it tests and it copies it and makes lots of copies. And if you've been exposed to COVID, it will then test positive because it will make a whole bunch of that sequence of DNA from that original RNA molecule from the virus. So if you've been exposed, then that DNA will show up in the test. Again, I'm not going to show these videos because I will post them on Schoology so you can watch them on your own. The next topic that we're going to talk about is DNA sequencing. And DNA sequencing is a process of figuring out all of the base pairs in the DNA molecule. And the Human Genome Project kind of sequenced the human genome. So back in 2003, they were able to successfully sequence the human genome, and they used DNA sequencing in a process very similar to the Sanger method. And the Sanger method is also known as dideoxy chain termination method. and it's a very complicated word, but basically what you're doing is the same thing that you do in the PCR machine, except you're using unique chain terminating nucleotides in addition to regular nucleotides. And so those chain terminating nucleotides are basically defective. And why they're defective is you can't add on another nucleotide when one of those nucleotides are added on. Those are also fluorescently labeled with different colors. So the A's and the T's and the C's and the G's are all going to be color-coded differently. And so when you put them into the machine, you're taking the DNA sequence, the DNA template that you're looking for to try and figure out the sequence of. You're adding the primers to get the process started. You're adding DNA polymerase. You're adding a whole bunch of regular nucleotides, so A's, T's, C's, and G's. But then you're also adding some of these fluorescently labeled defective nucleotides that you can't add another nucleotide onto, and you're putting them into a machine. And what's going to happen is it's going to make millions of copies of this DNA sequence. But each time it makes a copy, it's going to randomly pick up one of these defective nucleotides. And so at every single location, it's going to make a different length strand randomly. And so every time it makes a copy, sometimes it will pick up a defective one here. Sometimes the next one, it'll get one more letter, and so on and so on. And then if you arrange them by size, each last letter on those pieces are going to be color coded. And if you've got a piece that is every single base pair length, then you can figure out the DNA sequence. So the very first one, the shortest one, has a C on it, which tells you your template strand is a G. Then the next one is one base pair longer, and so it's going to have a G, and so that tells you the next letter is a C. And then the next piece is just one more letter longer, and one more, and one more, and one more, and so on. And so if you know the color code of those last letters, you can then figure out the sequence of the DNA strand. And so you're then putting it through this machine that reads those color-coded letters of every single length piece. And then that helps you to sequence the genome. And so that's how you can figure out the sequence of the letters and the bases in the DNA molecule. So that machine will then read it and tell you the letters, because it's arranging them longest to shortest, and it can tell you the letters longest to shortest, and then it tells you the sequence of the DNA molecule. So that's the general gist of DNA sequencing. Right, if we look at human genome therapy, what human genome therapy is it is when you are taking a gene, you're putting it into a vector, and typically the vector is going to be a virus, or oftentimes it's a viral vector, and it will then insert the gene into an organism. So we know that viruses carry DNA or RNA, and some viruses will insert the DNA into the host DNA. And so if we take the viral DNA out and we put the DNA that we're interested in into that virus, we can inject that virus into the people. They don't get sick, but the DNA that has the gene that we're interested in will get inserted into the DNA of that person. That person is then now producing the protein that they couldn't produce before, and so they can now make the DNA that they need. And so gene therapy is basically using a vector like a virus to insert DNA into a person's genome so that they can make a protein that they previously couldn't make. And the Johnson & Johnson COVID-19 vaccine is a delivery mechanism to insert a gene into a person. And so the Johnson & Johnson COVID-19 vaccine was actually a virus. They took the common cold virus and they put the gene for making the spike protein into that virus, and then that's what was injected into people so that we could then learn how to make that spike protein. So it had the gene for the spike protein. The cells were then making the spike protein, which is then launching our immune system to learn how to fight off the infection. The Pfizer BioNTech COVID-19 vaccine, that was a little bit different, not a viral vector. It was actually a little lipid bubble that had nanoparticles in it. So nanoparticles that kind of were lipid protein coated so that it could actually get into our cells, so our cells could then make the protein. And then the next topic that we're going to talk about real quick is CRISPR Cas9. And so we talked about restriction enzymes, how they cut the DNA. CRISPR Cas9 is another DNA cutting mechanism, but CRISPR Cas9 is a newer technique. And actually in 2020, the people who discovered CRISPR Cas9 in about, I think it was 2011, they figured out this whole CRISPR Cas9 thing. So not that long ago. It's a very new process, but it is better than the restriction enzymes because it's actually programmable. So it's a programmable editing tool that you can cut and edit the DNA however you want it to. So you can do all kinds of things with it. You can cut the DNA in a certain location to disable a gene. You can cut out a letter and change a letter. You can add a whole sequence of DNA to an organism with CRISPR-Cas9. So it is unique because it is a programmable editing tool. It has huge, widespread capabilities. And so scientists are doing all kinds of research and trying to do all kinds of therapies using CRISPR-Cas9. So that's the latest and greatest technique with that. Okay. COVID-19 vaccines, we're not going to actually watch those videos, but I will post this so that you can watch those if you are interested in watching that. So that kind of wraps up a brief talk on biotechnology, and let me know if you have any questions.